everyone, I'm the Liposome and I'm here to explain you my components and functions. I was first discovered in the 60s by the British hematologist Alec Bengham, who studying the hydrated phospholipids under an electron microscope noticed me. For the first time, the researchers discovered that the cell membrane is composed of a double layer of lipids. In fact, I'm a vesicle formed by a lipid bilayer, and in the center I enclose an aqueous compartment. Depending on the characteristics of the polar head groups of the amphiphilic groups that make up the double layer, 4 types of liposomes are distinguished: anionic, cationic, neutral, and zwitterionic. Anionic liposomes are characterized by a surface covered with negative charges and are able to surround DNA, transport it inside them, and convey it to cells. The liposome DNA complex is called lipoplex. Cationic liposomes, instead, have positive charges on their surface that can attract negatively charged DNA and keep it anchored to their surface, or can incorporate DNA to convey it to cells. The incorporation of lipoplex into cells can take place through 2 pathways: by direct fusion with the membrane, followed by the release of DNA into the cytoplasm, or by a process of endocytosis that depends on the formation of clathrin-coated vesicles. The second one is the predominant process. Once inside the endocytosis vesicles, DNA must escape from them, and if necessary, be transported to the nucleus. For the internalized DNA lipid complexes not to become trapped in the endosomes and to be subsequently degraded into the lysosomal compartment, endosomal escape must take place. That is a flip-flop mechanism consisting in the destruction of the 2 structures, namely the endosome and the liposome, and in the release of nucleic acids in the cytoplasm. One of the main applications of liposomes is drug delivery, which aims to locate the sending of active substances to the site of interest, such as a tumor or an inflammation. Advantages: they naturally complex with DNA and interact with cell membranes, are internalized by endocytosis and/or membrane fusion. DNA is released into the cytoplasm, protect DNA from cell degradation, biocompatibility, biodegradability, low toxicity. Detriments: weak stability, low encapsulation efficiency, the release of the load is fast, short biological half-life, initiate immune responses by activating the complement system.